Hello students, welcome to you all. Today's topic of discussion is anti-protozoal agents. In this topic, I will discuss introduction, classification, mode of action of anti-protozoal agents and synthesis of metronidazole. So in the countries having temperature zone, protozoal diseases are of minor importance, but bacterial and viral infections are widespread. On the other hand, protozoal diseases are common among people in underdeveloped tropical and subtropical countries, where sanitary conditions and hygienic practice and control of the vectors of transmission are inadequate. In some areas with temperature climates having poor sanitization conditions, the prevalence of amoebiasis has been estimated to be as high as 20% of the population. However, with increased war travel, protozoal diseases are no longer limited to any specific geographic areas. As we know, protozoal diseases are caused by protozoal cells who are unicellular eukaryotes. Protozoal cells have metabolic processes near to human hosts than to prokaryotic bacterial pathogens. That is why treatment of protozoal diseases becomes easier than bacterial diseases. But one of the major disadvantages of anti-protozoal drugs include serious side effects in the host, particularly on cells having high metabolic rate. Following table includes details of various protozoan diseases, causative agents or parasites and organs affected. First one, amoebiasis, which is a parasitic infection of the intestines caused by the protozoan Entamoeba stoltica. Organs affected are intestine and liver. Second one, African sleeping sickness is also known as African trypanosomiasis, and the causative microscopic parasites is of the species Trypanosoma brucei. It affects blood and brain. Next one is leishmaniasis, commonly named as Kala-Azar, is caused by Leishmania donovani, and it affects white blood cells, skin and intestine. Malaria is a mosquito-borne infectious disease that affects humans and other animals. Causative agent is Plasmodium species, and it infects liver and red blood cells. One more disease known as babesiosis is a life-threatening infection of the red blood cells that is usually spread by ticks, and causative agent is Babesia microti. Antiprotozoal agents or drugs represents a class of pharmaceuticals used in the treatment of protozoan infections. Classification of antiprotozoal agents include a number of drugs classified according to their structural properties. First category include nitroimidazole derivatives such as metronidazole, tinidazole, and ornidazole. Second category, dichloroacetamides include drugs such as diloxinide, itofamide, clefamide, and taclosan. Third category is of halogenated 8-hydroxyquinoline such as idoquinol. Fourth category, alkaloids include imetin and dihydroimetin. Last category is of miscellaneous agents such as pentamidine, isethionate, atovaquone, eflornithine, nifurtimox, benznidazole, milarsoprol, sodium stevogluconate, dimercaprol, and ceramine sodium. Besides these, antimalarial agents are also antiprotozoal agents. We already discussed antimalarial agents in previous lectures. Nitroimidazole derivatives are 5-nitro-substituted imidazole derivatives. Following are the structures of metronidazole, tinidazole, and ornidazole. Dichloroacetamides are chlorinated derivative of acetamide. Following structures are of diloxinide and itofamide. These drug candidates are used in the treatment of amoebiasis and some other protozoal infections. Oxine, quinophenol, or oxyquinoline is the parent compound from which the antiprotozoal oxyquinolines have been derived. Idoquinol is poorly absorbed from the gastrointestinal tract and is used as a luminal amebicide. The alkaloids emetine and dihydroemetine are obtained by separation from extracts of ipecac root. Last category is of miscellaneous agents. Following are the structures of pentamidine isothionate, atobaquone, and eflornithine. Next is mode of action of antiprotozoal agents. First of all, I will discuss mode of action of metronidazole. It is believed that metronidazole diffuses into the organism, inhibits protein synthesis by interacting with DNA and causing a loss of helical DNA structure and strained brachis. Therefore, it causes cell death in susceptible organisms. The mechanism of action of metronidazole occurs through a four-step process. In the step 1, metronidazole enters into the organism by diffusion across the cell membranes of anaerobic and aerobic pathogens. Step 2 involves reductive activation of the drug by intracellular transport proteins by altering the chemical structure of pyruvate ferredoxin oxidoreductase. Further, the reduction of metronidazole creates a concentration gradient in the cell that drives uptake of more drug and promotes cytotoxic free radical formation reaction. In the step 3, cytotoxic particles interacts with host cell DNA, resulting in DNA strand breakage. And in the last step, breakdown of cytotoxic products occur. Mode of action of diloxinide. Diloxinide is a luminal amoebicidal in nature. It destroys trophozoites of Entamoeba stoltica and prevents amoebic cyst formation. However, the exact mode of action of diloxinide is still unknown. Next is mode of action of emetine and dihydroemetine. Both emetine and dihydroemetine act on tissues trophozoites, causing irreversible block of protein synthesis by blocking movement of ribosomes along mRNA. Thus, it kills trophozoite forms of amoebas directly, but is not very active against cyst form of Entamoeba stoltica in the intestinal wall. Mode of action of atobaquone. Atobaquone is an inhibitor of parasite mitochondrial electron transport. It generally does so by targeting the cytochrome BC1 complex. It also believed to inhibit nucleic acid and adenosine triphosphate ATP synthesis in susceptible cells. Synthesis of metronidazole. Following synthesis involves three-step reaction. In the first step, glyoxyl is reacted with ammonia and acetaldehyde. These reagents upon cyclization reaction forms heterocyclic compound 2-methylimidazole. In the second step of the reaction, 2-methylimidazole undergoes nitration reaction in the presence of nitric acid and sulfuric acid and forms 2-methyl-5-nitroimidazole. In the third step, 2-methyl-5-nitroimidazole reacts with 2-chloroethanol in the presence of basic medium and finally forms metronidazole. Metronidazole is an antibiotic that is used to treat a wide variety of infections, such as to treat skin infections, and mouth infections. It is used in the treatment of conditions such as bacterial vaginosis and pelvic inflammatory disease. Metronidazole may also be used with other medications to treat certain stomach and intestinal ulcers caused by bacteria H. pylori. Thank you.